Okay, and thank you everyone for joining today. And my name is Deidre Dalmas. I am the scientific director of the investigational safety group within in vitro in vivo translation at GlaxoSmithKline. And today I'll be presenting about the application of complex in vitro models in the discovery and development and highlighting some aspects from an IQ-MPS landscape survey that was conducted in 2019 and 2021, and some of the differences and similarities between them. So I am presenting on behalf of the IQ-MPS survey working group. So currently, there are many opportunities for more predictive models earlier in research and development, and that has been something that has been ongoing for many years. For example, highlighting some work that was done in 2015 on the analysis of attrition across various different pharmaceutical companies, and really related to this is the overall concept of the lack of preclinical translation in the various different models that are currently being developed and the need for better predictive models. And, over the years, there's been, a lot of different work that has been ongoing to be able to help try to generate these better predictive models. And across this, some of the aspects of this, really incorporate trying to, reduce animal use, generating better screening models, using cell cultures, using various different types of platforms. And what you can see is, over the time, there's a large amount of time that is spent in the various different phases of clinical development. And so possibly, what we're looking at is really being able to develop better models that recapitulate and have better translation to the clinic to be able to help us, one, get patients medicines faster, as well as reduce the amount of time that we're trying to develop these molecules to get them to market. And along with this is, where possible, what we would like to be able to do is combine, safety and efficacy, predictive models within these concepts that we're trying to develop as we're moving forward. And so in doing so, what we really want to do is to be able to help increase our translation into the clinic. And what you can see is here on the right-hand side is the likelihood of approval from phase 1. And, over time, what you can see is that there's a various different, I would say, systems that where we have some weaknesses, translational weaknesses across the various different organ systems that we have. And that's one of the key features and one of the key areas where we really want to focus in the future is being able to adapt and use these alternative models to be able to help us be able to close that gap and us really be able to be more predictive for the future. And, over the past few years, there's, as everyone knows, there's various different reasons for failure as we're trying to move forward in drug development. And a lot of these occur preclinically, a lot of these occur in the clinic. And the reason for this slide, there was a study that was done in 2019, and that is from the Cancer Treatment Reviews, where this is pulled from. And what you can see is, just to highlight as we're moving forward for these alternative models, is that to highlight looking at, say, for example, the liver, which is number 3 there, which is kind of like the burgundy red sort of color. Both preclinically and clinically, there's still a lot of attrition within these areas. And what you can see, 14% preclinically and around 12% clinically. And that, really opens up the doors for us and shows the need that we do still need better predictive models in, both bases if we're going to be able to move forward. So bringing in, the alternative models and alternative methods that we can help address that is a need for the future. And then how can we do that, from preclinical success and helping to reduce that clinical attrition using these both, from a PD standpoint as well as from an efficacy standpoint as similar in safety assessment. And, part of this is really, choosing the right models and in the right endpoint for these types of studies. So as we move forward, what you would want to see is they're starting from the left to the right is that, we really want to be able to know our patient population, what we're looking for, and, what we're trying to model in these systems. And in doing so, we really have to understand what the right complex in vitro models or microphysiological systems are that are going to recapitulate specifically what will be, I guess, most reflective of what that clinical disease state potentially is to be able to move forward properly. So, some of those different types of platforms that, we're talking about from a standpoint of alternative models is really those complex in vitro models and microphysiological systems. So whether it comes from and how they're developed, such as bioprinting tissue, scaffolds, organoids, various different spheroids, and how those tissue systems can be used to the best advantage to be able to help us move forward into the future. And then, once we identify what those proper systems are, or the best systems that we feel recapitulate what we're trying to achieve and that clinical system, whether it be from the animal standpoint or as we move into the human standpoint, and what is best translatable in that area is really understanding and ensuring that these complex in vitro model systems or microphysiological systems really are able to recapitulate the right pharmacokinetic parameters. They have the right physiology. They can potentially predict the right toxicity to be able to be applicable to the assessment of safety as we're moving forward. And some of the ways that we can do this is really, with these systems and having the right cells and the right characteristics, is then being able to again use them both from a pharmacodynamic perspective and an efficacy perspective, as well as from a drug safety assessment perspective. So for example, looking at tumor identity and trying to ensure that the molecules will be able to be I would say applied into these complex, in vitro model type systems, and really truly predict, potentially, the clinical outcome and how they would behave pharmacodynamically. And if we see efficacy as well as from a drug safety assessment standpoint. So for example, liver toxicity, could that be something that can be recapitulated, in vitro in these alternative solutions and alternative models that we're trying to develop to be able to do that these days. And in that same sense is really applying and utilizing these systems. So if they do recapitulate, the clinical environment, and really looking and determining can these systems also be used to assess, say, for example, safety biomarkers. So, we have a lot of safety biomarkers. We have a long way to go in many of the various different systems, but if these models can be used for that, that will not only help inform, the clinic and ensure we have the right population of individuals as well as the right endpoints, and in the same sense, also helping to reduce animal use as we move forward. So really what the overall goal there is to be able to use these platforms, such as, these in vitro, complex in vitro systems and microphysiological systems with flow and, involved, is to be able to really try to identify what that therapeutic index is and be able to predict that in a very, much more efficient way than we are currently able to do today. So how do we adopt, using these more predictive type 3D in vitro models for, drug development as well as for safety assessment and, PD assessments and efficacy is, right now what we really need to do is, build confidence in these models. There's a lot of work that is ongoing in this space, across the, academic, across the pharmaceutical industries. And a lot of vendors are trying to develop really translational models, and what do we need to be able to do to have confidence in moving forward, and how sensitive can these models really be. And with the overall goal is that we really want to have these models have translational relevance. We don't want to just have them, okay, yes, there's a a predictive endpoint or sorry, like a, pharmacological or a cellular response that, isn't going to translate to something, clinically. Because we all know that, there's many examples of where, we can take an in vitro culture system of, a single cell, type model, and we can see changes, but then what relevance does it have, clinically? So really we need to understand what and identify what the value is in generating and this data and what that critical data is to be able to use these models, and also in ensuring how we are going to use these models from, say, the context of use. Are we looking for pharmacodynamic markers in them? Are we looking for safety markers? Are we looking for biomarkers? And how can we go about doing this operationally? It is a, very expensive and time consuming process to be able to develop these models, ensure we have the right cells in them, and ensure that these cells are interacting, properly to be able to recapitulate what would be expected. There's, again, a lot of individuals that are working in this area, and some companies are doing this in-house, some are doing it externally. And really the need to be able to use these microphysiological models that really do recapitulate pharmacokinetics. I would say ADME and others is really having those, individuals who are, experts in that area, being able to apply their, expertise, I would say, um, mm, to be able to recapitulate and assess from, the standpoint of biology as to, really how can we move these molecules and these models forward to be able to use. So, some of the key areas that are considered, across the various different organizations, that I've been, part of listening and, participating in conversations, is really using these for target validation. Are they a high throughput system? Are they more a medium throughput system? How are we using these? Are we using them early in the drug discovery and development process? Or, are we, using these more from a context of use and qualification, say, of, say, biomarkers, per se, or qualification of certain pharmacodynamic or efficacy endpoints. So this all really, pulls into that, safety assessment, because that whole process is starting from discovery all the way through development, and, encompassing that safety assessment. So where do these models fit in? And, there can be various different contexts of use for these. So understanding where each model and what the, benefits and the, I would say, the gaps are in them, will be able to help us, move forward. So just looking at incorporation of the complex in vitro models into this process, and how this is, moving forward, is that, obviously, you have the patient with the disease, you have organ tissues, we understand, from the standpoint of, an individual, what potentially is going on. And from an intracellular standpoint, in some instances we don't. And, there's various different organ systems, that are key moving forward, and that is generally the highest number of models that are kind of getting generated there, right now, from, whether it be from the liver, from the heart, from the kidney. And, there's a lot of activity in this space, and, really taking, these organ systems and being able to, isolate and extract, say, potential cells that will be able to be a model, something that is more disease relevant, so that we can, move forward and help one, going from the bottom to the top, screening and helping to identify target identification and validation. Moving forward, can you utilize these systems to be able to help, identify the compounds, to edit genes, to be able to see what would happen if we did that. And then, as we move forward through the drug development process, it's really looking at the larger, compound screens. And, once you get into the area where, you've identified your compounds and moving forward, that's where, we want to be able to determine, can these systems be used, from an efficacy, a PD standpoint, and be used for, safety assessment and beyond. And looking at them, like, as I said, mentioned before, we're looking at PK, PD. Can we predict doses in the clinic? Can we predict what the safety will be from the various different models? And, there are a lot of different manufacturers out there. And, where we are in the moment is, and moving forward is can these systems, once we get them to the point where we understand what they are, once there's a good sense across the industry as to that they can recapitulate in many instances, they can at the moment, but how can they be used from a drug development process and safety assessment process, and that's potentially evaluating, as I said before, looking at new or identifying new potentially biomarkers in this space if these systems really do recapitulate the clinical aspects of them and what would be expected, looking at safety biomarkers. And this data can be used, we're starting to gather tons and tons of information, these large big data sets, whether it be from safety, from toxicogenomics, or other endpoints, and be able to utilize this information more in a QST model approach, where we can, take this information, such as, PSTC is doing that as we're moving forward and trying to gather, the information from, say, potentially clinical studies, from non-clinical studies, and trying to pull this information together to be able to better predict, one from a safety standpoint, biomarker standpoint. And then similarly, once we have these models developed, going in and taking these this data and then applying it to those patient populations, and being able to assay, not only the complex in vitro models for such as imaging, flow cytometry, cytokine profiling, and again, biomarkers, and how can we potentially use this going forward. So with that being said, I am a part of and a member of the IQ Microphysiological Systems Consortia. So, and with that, the IQ-MPS affiliate that is part of the IQ Consortia has done some work in this area, and I guess working to uh, ask across the industry some various different questions. So before I do that, I just wanted to go over a little bit of what the IQ mission is for that. And really, it is to advance science and technology to augment the capability of member companies to develop transformational solutions that benefit patients, regulators, and the broader R&D community. And IQ does have various different working groups and different subgroups underneath that. And the IQ Microphysiological Systems affiliate is one of those. And the main goal of this is to be a leader for both MPS developers and the various different stakeholders across the industry for the qualification of microphysiological systems and models. And it also is a way to have a pre-competitive cross-pharma collaborations and data sharing to help facilitate the implementation of the use of these models, both for qualification and for use. And there is a generally a focused engagement between industry and the regulatory agencies on the current status and the evolving field of this area. And there's a lot of discussions that are ongoing between the both of these groups. So it's a a great collaborative effort to be able to help move this area of field forward. And then obviously, what a goal as well is to develop external partnerships to be able to, enhance the inclusion of industry, across the various different models that are developed, and to be able to hopefully develop something that, these models that will be clinically translatable. So with the IQ-MPS membership, there's 21 different members currently within that organization, and they are all listed here. I won't go over each and every single one of them. There is diverse representation from safety, from the 3 Rs, from the ADME part of IQ, as well as covering PK and PD. And so there's a lot of individuals that are involved in collaborating in this area for these complex in vitro models. And the leadership is listed here, and they are very cooperative and collaborative, and are really driving forward the use of MPS models for both for drug development as well as for safety assessment. So part of the MPS affiliate that and working group that I'm involved in is was pulling together the various different organizations and really developing a collaborative questionnaire that was, circulated. And, within the MPS area, there is a definition that is utilized for these activities and the various different models, and that is that it defines microphysiological systems as going beyond traditional 2D culture models by including several of the following design aspects. And that really includes various different primary or immortalized cell lines. It can include micropattern co-culture systems, spheroids, organoids. It also includes bioprinted tissues. And then as we get, more complicated, is including, various different flow to these types of organ systems where, we really want to recapitulate, what is happening in that in vivo, system. And then, with that, as well as really generating cultures with various different types of molecules and interactions, such as, immune system components within these systems. So there is a very specific, definition as to, what this means. And as you increase in the complexity of, adding more cellular, types of uh, molecules to be able to really recapitulate that, and, adding fluid flow into that system too is very important. And so part of the survey working group of the MPS landscape surveys is what we set out to do. And there was a survey in 2019, and then we had another survey in 2021, which was earlier this year. So sometime, I believe, it was between, February and March of this year. So it was really to look back and to try to understand, where things are going, where things have come, and where things have moved over the last couple of years, because this is a very, quickly advancing area within, drug development and safety assessment as well. And really to better understand these surveys on how pharmaceutical companies are currently utilizing these complex in vitro models, as well as these microphysiological systems. And what are the major challenges to realizing the full potential of this type of technology? And the surveys that were conducted over, 2019 and then again in 2021 really focus on the various different platform and design aspects that, I would say, slightly differed between the two surveys. So keeping that in mind, in 2019, the focus was generally on complex in vitro models. And complex in vitro models, in the sense of that survey, really encompassed all of the, generally all of the, I would say, at the bottom of the screen, the various different models or platforms that you can see there. So micropatterns co-cultures, 3D microtissues, bioprinted tissues, and including, microfluidic systems and organs on a chip. That were, are currently being developed and were being developed at that time. And then in 2021, because there was such a, advance in this area, the survey in 2021 did, ask similar questions as to 2019. However, the focus was a little bit more on the microphysiological systems. And what you can see is, based on the 2021 survey, this really focused on models that have a flow component to them. So really trying to focus more on how people are using, those types of systems currently at this time. So based on what you can see there, the check mark means is the platform in the questionnaire included those types of design aspects, whereas the X, included that these were models that were not really considered in the survey responses. It does not mean that these things are not currently being utilized by the companies, but just that the questions that were asked really had a specific focus. And in the 2019 survey, there was 25 respondents from across the various different, companies, and whether it's large, small, and then or other. And then in 2021, we had about 25. We had 25 respondents to the survey. And this is really highlights from the survey. There will be additional, communications in the future. There were quite a lot of questions, so this is really highlighting, some of the just, high level results from that survey. and more to come in that area. So, based off of the various different company demographics that, the survey respondents were coming from, what you could see is, the question is, which of the following best describes the size of your company? So there's large, mid-size, and small companies. And what you can see is, that based off of the large companies, there are, more individuals, responding in 2021 versus, 2019, and then all the way down to mid-size and to, smaller companies. And, generally over the time and with the respondents, they were very similar across 2019 and 2020. And then, so just some of the highlights from the 2019 landscape survey was that, complex in vitro models, are applied, across by various different companies and are utilized for safety, ADME, as well as pharmacology. And, at the time of this survey, they were segmented across the various different groups. There were various different sources of complex in vitro models, and that is still true, true today. And these sources of these models come from vendors, academia, and internal to whatever company, whether it be pharmaceutical or small biotech companies. And then when looking at evaluating the types of models that they're looking and the type of species. And, in 2019, this was really focused on, human and clinical models. And again, this is similar, today with generally, I would say just from, perspective is that, there is more focus now on, moving forward with, the clinical models with, some questions I would say that's come up in recent, symposiums and, presentations that I've seen with, IQ-MPS, with IQ, others with the FDA on, the utility of also looking at the non-clinical species and how do these model, because there is both preclinical and non-clinical and clinical attrition. And a lot of the issues there are that we don't understand how those models translate. So if they can be translatable across, then we'll be able to, from a safety assessment perspective, understand a bit more on if we do see something within those non-clinical species, is it translatable or not, hopefully in the future. And then, in 2019, there was a limited interest in looking at multi-organ integrated systems, and, that's not shown here within this survey. However, as I'll go through in the next couple of slides, that is a little bit different now today based on the updated survey. And then looking at, in 2019, where these were used across the various different modalities, predominantly it was in, small molecules, biotherapeutics, and then some additional characterization and internal experience with these models was determined to really be necessary to be able to increase the utilization of these and apply this technology. And I think, based off of, my opinion is, based off of where we are today, that is still something that, and, is, a key, aspect of being able to utilize these alternative models in, drug development and safety assessment, and how do we really apply this technology to be able to help us move forward. And then before I go over some of the results from the 2021 landscape survey, is that, based off of looking back and which we will show, is that the interest in the various different organ systems, the main organ systems, was similar really between 2019 and 2021. And I will highlight this in the next couple of slides. And like I said just moments ago, whereas in 2019, there wasn't like a great, I would say, interest in looking at multi-organ systems. And, potentially this was, my thoughts was because, we, at that time, the area is moving very quickly, but people were just getting used to, and individuals and companies were just getting used to starting to utilize those systems. So now with the utility and the I would say success in various different models in that area, is that there is now an interest and in using multi-organ integrated systems, especially in the area in combination with, liver, the immune system, just, based off of, the complex, I would say, disease processes that we do have now and are being, understand that it's not just, one cell type that we're targeting. We're targeting, systems in and of themselves. So, um, generally some of the highlights of this are also, how do people use these? And I will go over that shortly. And then, where we are in the current, regulatory, state of, this. So one of the questions on the survey was, has your company used microphysiological systems and, complex in vitro models in the last 5 years? And what you can see from 2019 to, 2021, the number of survey respondents are listed there. And, at first glance, you look at it and you think, okay, well, why is there less in 2021 versus, 2019? However, please keep in mind that, the survey was very focused on complex in vitro models, and the 2021 survey is really focused on the microphysiological systems. And one can imagine that as the systems are becoming to get more and more complicated, it is requiring more and more skilled level of, individuals who can utilize these systems. And even internally, I would say within companies or within the vendors, is it's just a lot more complicated now. There's so many different models, and to be able to use that, I think we really do. There's a little bit more effort that goes into being able to utilize these molecules. And what you can see is that, there's just answering no, have you used these in the last 5 years? I think, as I said, the models are becoming more complicated, and, some, companies potentially, are being able to, are not focusing on potentially that area, but, really truly focusing on, that 80% there in 2021, indicating that these systems, are currently being used. So what was interesting really about the survey between 2019 and 2021 and was the organs of interest. And what you can see across this period of time, about 1920 was around 3 years there. I mean, really the first 4 and even, five-ish, systems that are looking to rank across them are very similar across, both years, whether you're looking at complex in vitro models or microphysiological systems, and, they're intertwined, just putting, that flow component to there to 2021. And, with liver obviously being, the most highly ranked organ, followed by the GI, kidney, CNS, immune system, and then the cardiovascular, system. There are a lot of, a lot of systems that are out there, and a lot of models for the cardiovascular system, and whereas there is, more lacking from some of the other systems. So just to kind of keep that in mind, is that hasn't changed really over time. And, the survey committee thought that that was a very interesting, aspect of both of those systems. And then in 2021, as I mentioned before, the use of multi-organ systems, both in, 2019 and 2021, there was, use of these systems, and, wanting to be able to have this multi-organ, integrated system. And what you can see, over there on the right-hand side is that, again, what you can see is that, liver is a very, active, I would say, proponent and active part of this work. And, there's a lot of interest in looking at the various different systems in combination with the liver. Similar with the immune system, what you can see is, that similarly, the immune system plays a large, component of, all of the different organ systems, and there is a large interest across the industry. And the count that it's listed there is that, the number of, you know, responses that there were in regards to wanting to be able to look at combinations of interests, in the future in those particular areas. And, what really does stand out about this is, that liver gut is, one of the highest ranked, the highest count there based off of the survey respondents. And then, across the bottom, there are various different other organ systems such as bone marrow, kidney, heart, skin, tumor, that, there are other individuals that do have interests in this area. So, moving forward, hopefully, we can begin to be able to explore these types of interactions within those systems. And then, so one of the other questions are how MPS data are used within the various different companies. And what you can see and what's interesting about this is that, in both years, the use of these to qualify, models for internal decision making, I would say a slightly lower percentage in 2019, and that's increasing in 2021, also keeping in mind that 2021 is the use of these microphysiological type systems with a flow component. So, that is an increase in those types of numbers because the, 2019 survey wasn't specifically focused on that. But, what is interesting in respect to, the answer to the survey question is that in, 2019, there were a question about, have you been using these, I would say, as part of this, question in regulatory submissions. And what you can see is that there was approximately about 19%. And then today, in 2021, based off of the survey, there is actually less than that. And, that can potentially be, related to the fact that, just the questions that were asked on the survey, but these questions, like these models are getting more and more complicated. It's not understood what if they were submitted and used in submissions, what would be done with that data, how would that reflect back on a question or a molecule that was currently in development there. And so I think there's some of the things that hopefully we'll talk about in the discussion part of this is like, how do we move forward with these systems? What is really needed by the various different companies to be able to begin to utilize those. And I think in 2019, there, are simpler systems. And I think, I don't think that it's gone from, 19% to nothing. It's, really, I think that is still, moving forward. It's just keeping in mind that the questions that there's just really focused on, microphysiological systems. I'm sorry. So as we move forward, just as I was indicating, there are some key unmet needs in this area, and really what information do we need to be able to have confidence in these models across the various different platforms? What are the contexts of use, and what is the qualification that is needed to be able to confidently use these? I would say both for qualifying the model for model development, for internal decision making, as well as for regulatory submissions. And what kind of considerations do we have to think about as we're moving forward, both from research and development, as well as again in these regulatory submissions. And as I mentioned before, there is that question, we really do want to have that translation. And some of our attrition does come from the fact that we do see I would say toxicity in the various different, in vivo animal models with the, not knowing in some instances if they're not well-known biomarkers or biomarkers in that area as to what would really be translatable. So being able to have these translate and, do you need that in a complex in vitro, type model system? Do you need that translation across them? Or do you focus on, the human systems without that, I would say without that link there. All right, so some of the key components, as I mentioned, that potentially could be considered, when we're developing these types of models. There was a paper that was Jason Eckert and Claire Zhang put out in 2018 on some of the aspects of this, and just really not going over this, one by one, but just keeping that in mind as we're thinking about utilizing, these alternative systems for safety assessment. What kind of path, what kind of assessment would be done to be able to, I would say, qualify them, to evaluate them, to ensure they have the right physiology. What kind of context of use would be needed, and then, how would we really validate these systems? And then, really the overall, goal of this is really trying to recognize that these alternative models and complex in vitro models hopefully can be transformative, and the potential and the power that these systems do hold within themselves. So how can we use them, some of the in an integrated fashion where you can have the greatest impact for disposition, safety assessment, as well as efficacy, and trying to move these really these models forward. And what are some of the goals, and keeping that in mind is that you really want to understand what the goals are of the model that you're trying to develop, and what area can it cross the various different areas. And there's a lot going on right now in the disease modeling, and hoping to be able to and applying the ADME to those types of molecules. Can these moving forward, can these systems again be used to evaluate and either identify or assess various different, maybe known or novel or new safety biomarkers as we're moving forward before we get into the clinic, to be able to help with that translation. Some of the questions as what qualifications are needed in these various different areas, and with again the overall long-term goal of trying to help reduce animal use as well. So as we move forward, really, these are the key, take-home messages for these alternative models is really, what is the cellular input into in the models? How do we improve the reproducibility of them? If someone, and I'll just throw some questions up there, if, a model is developed, with one company, and, it's potentially qualified, it, is looking good, does that mean that that model always has to be used? Is it that you need to, qualify just various different endpoints across the various different models? And so there's just all, questions that I think that, we have, and, folks have in this area. And, moving forward, there was quite a lot of questions that were asked on the MPS survey. So more of that information will be coming out in, the near future as to, the answers to some of those questions. So this, what I would shared was just a brief highlight. So, and again, the context of use, how are we going to use this? What specific mechanisms are we targeting? Are we looking at, you know, cross-species comparisons, looking at clinical dose predictions and potential safety concerns based off of these types of models? Again, species, translation, do you do forward and reverse translation? Can we utilize these models for those particular purposes? Expanding, the use of them we currently have right now. And then I think, what's on, most, everyone's, minds are from a regulatory aspect. How does this come into play as we are moving forward and developing in this area? And there were some questions that were included in the IQ-MPS survey around these aspects. So they'll be coming out in in the future as to what the industry and others, think in this particular space. But, generally, some of those questions, and, for, our discussion later today would be, do we need specific guidelines in this area? What's currently being developed? How do we interpret that data? What are individuals thinking in response to that? If you submit, some of this data, how will the regulatory agencies look at it? Will they come to different conclusions than what you have? So again, they're all various different questions that I think will again come into play as we're moving forward in this area. And then with that, I just wanted to acknowledge the various different individuals across the IQ-MPS survey team. Tom Baker is the chair of that group, and Sylvie Chaco have been very involved, as well as all the other members of that survey team, and are currently in the process of going through all of the survey results. So again, what was shared here are some highlights from that. And then also the IQ-MPS leadership and the affiliates, and all those individuals who either participated in helping to review the survey before it was sent out, all the affiliates members who responded to the survey, the communications team, and everyone else at IQ that was involved in generating that survey, as well as being able to review this presentation for review. So with that, I just wanted to thank everybody for your attention. There will be, some additional survey responses in the near future, that you'll see through various different presentations, publications potentially. And so keep your eye out for that. And I know there are, we'll hold questions until, we have our discussion. So thank you for your attention.